So let's move on to other environmental compartments with the presentation of our second speaker. We are pleased to welcome Dr. Gwenaël Imfeld, CNRS Research Director at the Institut Earth and Environment in Strasbourg, ITES, which is a joint research unit between the CNRS, the University of Strasbourg, and the National School for Water Engineering and the Environment. He is the head of the Research Federation on Environment and Sustainability in Strasbourg. So Gwenaël is an environmental biogeochemist specialized in pollutant transformation and microbial ecology within continental hydrosystems. And he worked on reactive transport of micropollutants, and to get a better insight into their fate in soil and water bodies, he combined isotopic, hydrologic, and biomolecular approaches. So he is going to talk about the fate and impact of micropollutants on ecosystem and human health, taking into account toxic effect in OneHealth frameworks. So you have the floor.
Thank you very much, Stephanie. Thank you, everybody, to all the organizers and to everybody. So I'm very happy here to present some insights on the micropollutants in the environment. Just like for CO2, we also have a micropollutant footprint as human beings. What I would like to do within the next minutes is to present you some interdisciplinary environmental research on micropollutants, to give you some insights on the forefronts of this research. And I will try then to connect this research to the OneHealth actions that can be taken. OneHealth is an attempt to connect the animal, the human, and the ecosystem health. And this is extremely challenging. And I would like to show that ecosystem health is still not totally in the OneHealth concept. So first of all, I think it's very important to remind that actually the gross domestic product is equal to a production of goods and services, but also at the price of huge wastes, emissions and pollutions. So if you would like to reduce the wastes, indeed, we need to change the growth or to improve, as we have seen yesterday, through a full circular economy, the production in order to reduce the waste. Regarding chemicals, over the last years, the number of registered chemicals have increased by 5 times from 2005 to 2018, and the sale value of chemicals will increase by 3 times from 2010 to 2050. We also have to keep in mind that the chemical pollution causes about 9 million premature deaths yearly. Actually, micropollutants and their transformation products are novel entities. And you can see here this concept of planetary boundary, with here in green the safe operating space. And here we were talking about climate change. This is just one of those different boundaries. And one other one, which has been now quantified to be overpassed last year by Pearson, is the novel entity. The characteristics of those novel entity is their persistence, their mobility, and their potential negative impacts on the different health. The planetary boundaries are transgressed based on their increasing rates of production and release into the environments and toward the different reservoirs. The higher release, the novel entities with diverse risks and a huge diversity of different chemicals. And also our ability to conduct safely the assessments and the monitoring of those contaminants has been exceeded. So that means we are producing so much compounds which are themselves degrading in transformation products, giving themselves transformation products that we are not able to monitor them. Actually, micropollutants represent a huge amount of different compounds. Plasticides, colorants, flame retardants, pharmaceuticals and personal care products, and others. They come from different sources, agricultural also from the urban areas through the wastewater treatment plants, and they are gathering in water bodies or in air. And here, this is represented a water body where the different contaminants will accumulate, making here diverse potential impacts on the organisms, the humans, on non-humans entities. And you have here the different connections within groundwater, but also with particles. And there are actually 4 main domains of research for micropollutant research. The first one is the fate and the transformation products, to understand the behavior of those micropollutants. The second one is the effects on ecosystems, organisms, how they live, how they will evolve, especially given the cocktail of pollutants. Another important field, this is the human health and the impact of micropollutants on human health. And of course, the regulatory framework, how to evaluate the risks and assess the priority compounds to take action. An example regarding the fate of contaminants is the biocides. Biocides are found in huge amount in daily life material. Here, as an example, the building facades, the paints and renders. Do you know that in almost all the paints, the outdoor paints here, they contain biocides? Among them, terbutrine. Terbutrine is a triazine that has been forbidden in agriculture 30 years ago, and that it is now used in quite huge amounts in facades. Very few people know it. So we carried out with sociologists some survey in the city of Strasbourg, but also in other cities in Germany, close by, in Freiburg. And we could demonstrate here that consumers, city services, and professionals are not aware of those micropollutants found in the facades to protect them against the growth of algae and so on. Basically, those substances, they aim to keep the facades white, because white is good, white is clean, and green is bad. So there are also alternatives that exist, of course, mineral paints, sustainable buildings, but those alternatives are usually not known. So we carried out some experiments. Here you have outdoor facades. Those outdoor facades were exposed to the environment, to the sun, to the rain, to evaluate the leaching of terbutrine and their transformation products. So you can see here, terbutrine is found within the facades, will be here released at the surface of the facade, and you have here this molecule, terbutrine. And over time, with the sun and so on, we could identify a huge cohort of different compounds, about 20 of them, here the 4 merged ones. So you can see transformation products are about the same molecules, but with some cleavage at different points of the molecules. What we could show here is that transformation products are released to a larger extent than the biocide itself, that terbutrine itself. So this means here that unknown, potentially toxic transformation products are constantly released in higher proportion than the parent compounds. And this is not very well known. Regarding now the risks on ecosystems, we carried out here a study on terbutrine. I was just talking about metolachlor, which is a herbicide, which is forbidden this year on maize and sunflowers, and metformin, which is an anti-diabetic pharmaceuticals. And all these are then converging in aquatic ecosystems where they are found mainly in the sediments. So we evaluated here the impacts on microorganisms, because microorganisms are really the background of all the biogeochemical cycles in the environment. We have here mimicked different water sediment interfaces with non-spiked, single-spiked microcosms and microcosms spiked with the mixtures. And what we could find here, actually, is that these micropollutants in mixtures, they have no impacts on the kinetics. So the rate, the degradation is the same. However, we could observe that the microbiology, the microbial communities are shaped by the presence and the occurrence of mixtures of pollutants in a different way than with the individual micropollutants. And we could also observe that metolachlor and terbutrin were mainly shaping the community. Another example on the micropollutant impact, this time on the human health, is the co-occurrence of antidepressants and antibiotics in wastewater treatment plants. They are always found together, and this is only since a few years that research is ongoing to understand what is the effects of this combination. Actually, antidepressants induce antibiotic resistance and persistence, which can have, of course, quite important consequences on the human health. The presence of antidepressants will enhance the reactive oxygen species at the cell level, the stress signature response, and stimulate the efflux pumps through this increased antibiotic and increase then the antibiotic resistance and persistence. So you can see here it has a direct impact in the presence of antidepressant on the bacteria. Okay, regarding now the regulation, to improve the regulatory framework, it is not so easy because there are quite few cases which are extremely well documented. So you can see here, each dot represents a case study, and micropollutant subgroups with the highest priority could be identified based on the risk quotient. And you can see here the contribution of the different types of micropollutants to these risks. It's also possible here to classify by risk priority the different compounds. You can see here that, for example, pharmaceutical and personal care products play a quite important role and have an important position within this priority list. Such integrated approach are actually crossing the risk intensity and the frequency of micropollutants to determine their priority. It's quite useful for the regulation because it's today impossible to target on all micropollutants. Of course, an important point, is to think at the source. What I showed before is the interdisciplinary research, but nothing will here be superior or more advantageous than the prevention at source. And this is the interdisciplinary grand challenge in this field, going towards a full circular economy designed to prevent waste and micropollution from the outset. So before using a micropollutant, how the design based on the real social needs and the life cycle thinkings could be improved. That's a real key question. It's not only to have the regulatory framework stating the risks of these compounds, it's very important to understand, do we really need it, and what will be the life cycle of those compounds. So within this full circular economy, the pollution prevention is considered at every design stage to minimize the negative impacts. So from the sourcing to the manufacturing, we should rethink, reduce the waste that are produced, and when those wastes cannot be avoided, how to remanufacture and recycle. Of course, when we talk about micropollutants, it's very difficult, but we could think, for example, on antibiotics to avoid putting them into the environment. That would mean, for example, reconcentrating them from the urine directly. And we do not have to be shocked about those kind of things. This is possibly the future, because only 2/3 or 1/3 of the antibiotics will be effectively used in our body. 2/3 will be rejected in the wastewater treatment, and wastewater treatment plants are not efficient to clean those compounds. And this, of course, opens quite a lot of new research fields, like non-polluting components and process during the life cycles, how to include the circular economy in regular frameworks, not only the risk and the toxicity, the cost estimates of micropollutants. This is a very good point also to communicate on micropollutants, for example, through the labeling. When people, they understand that it costs some things to the society, it's also a good way to prevent, to think about it, and how to eliminate the most toxic micropollutants, residues directly at source. A key point here is that any incremental chemical or technical innovations will postpone the challenges to the future, which are postponing the challenges to the future, are actually dystopic solutions. Every single innovations that do not take into account this full circular economy is to some extent dystopic, because it's simply postponing to the future what could be thought at the real beginning. And this is, I would say, probably also a difference between the green chemistry, which has a lot of advantages, compared to the sustainable chemistry. Okay, it's moving a little bit, yeah. Another key challenge here is to integrate the increased toxic effects within OneHealth frameworks. So when crossing the planetary boundaries and increasing the human populations, of course it will impact the global changes and human activities. This will affect the increase of toxic risks, also through the multiple release of a huge diversity of micropollutants. This will directly affect the ecological equilibriums and also generate loss in the biodiversity. This loss in biodiversity through the destructions of the habitats, but also through the enhanced contacts between livestock, human, and the wildlife will increase the infectious risks. The increased toxic risk will also have an impact on the immune or endocrine systems of human bodies and other organisms, and will also alter the biotic interactions. For example, the relationship between the hosts and the pathogens by affecting the pathogens. And this all is enhancing potential chronic and non-chronic diseases. A typical example of this is the antibiotic resistance, antimicrobial resistance. Antibiotics are sometimes overused, and they will go in reservoirs, in animal reservoirs, in the livestock, and in humans, and end up in the agricultural reservoirs. So this is the traditional cycles, and then it can come back here through the agriculture, the food, back to the human reservoirs and the livestock. So the genetic materials and the bacteria will move in those reservoirs. But what happens if we add here the ecosystem health? That means here agricultural systems and livestock are connected actually to the ecosystems. And sometimes this is a little bit missed out. If now we add again the global change, this raised new questions which are overlooked. How global change will affect the spread of antimicrobial resistance, how antimicrobial resistance is connected to the biodiversity alteration, how climate change will affect the agricultural type, and through the agricultural type, the antimicrobial resistance, how micropollutant cocktail will affect antimicrobial resistance and persistence. These are their questions for the future, connecting these traditional veterinary and human health systems together with the ecosystem health. So chemical toxic risks are implicated on many levels of OneHealth, but there is still marginal incorporation of micropollutants and environmental quality in the large sense into OneHealth framework. And the question is why? And I would say there are two main ceilings, plafond verre here, glass ceilings to break. And this should come from the interdisciplinary research on micropollutants. So this is the first reasons I will just discuss. And the second one is going towards harmonized framework between ecosystem health and OneHealth. So the traditional research on micropollutants is trying to generate shared indicator on the micropollutant phase, the equilibrium dynamics of perturbated ecosystems. With those shared indicators, we're able to move and to evaluate toxic risks and the response of organisms and ecosystems, sometimes functions, and evaluate the risks. And here comes our first ceiling. Yeah, this is the first glass ceiling. This is the response. Some research are doing it, but it's sometimes not included at the very beginning, is the procedure for adaptation to mitigation and risk exposure. What will be the procedure with those findings? How do we do? The second ceiling, the second glass ceiling is the famous someone should. Okay, this is my part, and as a scientist, I can now stop. Nobody will do it. So, a little bit like the micropollutant in the full circular economy, at the very beginning of our research, one should see what could be done here at this stage to break this ceiling. This is the contribution to the socio-political decision, making process and implementations. So to think how this research can then contribute to this, monitoring the effects, and of course communicating, thinking about the data banking, the transfer of best management practice. And this is the sharing and operating innovative solution phase of the research, which requires non-academic expertise and actors. And this is where interdisciplinary research on micropollutants should move towards transdisciplinarity for OneHealth. So a typical framework toward OneHealth that includes toxic risk of micropollutants should be seen at different levels. This is first a matter of dialogue. These are the traditional discipline, epidemiology, physiology, toxicology, in human and animal health. The question is how to strengthen other disciplines in environmental science, which are extremely important to understand tomorrow's and today's challenges on the ecology, evolutionary, and environmental sciences. Here, now we can move towards the multidisciplinary stage. Multidisciplinary stage, that means here that you have a common umbrella. A common umbrella in this framework could be the multifactorial and non-communicable diseases. How transdisciplinary, or how disciplines such as eco-epidemiology, ecophysiology, or ecotoxicology could be strengthened in this framework and give all the place to those disciplines, linking ecosystem health with the other types of health. Then we have the participatory research, and this is very important. It's still not transdisciplinary, but here you can see the non-academic partners, the non-academic knowledge, share knowledge and discuss with the academic knowledge. And here the goal is to break down the sectoral partitioning and maybe also to think about the co-training, bringing together the communities to organizing, I would say, regular spaces for exchanges and also for strategic thinking. This would also allow the research to start from the usage to design the research. What is actually the usage of my research? Then moving to interdisciplinary, this is a steps where knowledge can be integrated and networks can be done within and among disciplines. For example, to target the micropollutants and the pathogen systems. Here, there is a bunch of research that can be done on toxic exposure and impact on immune and endocrine systems, multiple chronic stresses and response at the individual, population, or the community levels. Also, the epigenetic basis of population response to toxicants. And the transdisciplinary is here to frame a common goal between academic and non-academic knowledge, not for science, not for society, but with society. To build here integrated framework of health, agriculture, and ecosystems. That should be, of course, include some coordination, screening networks, databases, citizen actions, but also to include those issues, socio-economical, social justice, and regulatory. The goal of this transdisciplinary research is to build decision-making tools on the one hand, but also to define the future strategies. An example of this is here the Interact project that we are currently launching together with our German colleagues. We have here included the city of Landau, Freiburg in Germany, together with the Eurometropole of Strasbourg and the water agencies. The goal here, and the common goal, is to go towards a biocide-free city. How to eliminate building biocides and PFAS at source, evaluate antimicrobial resistance and reduce antibiotics. This is together also with sociologists and anthropologists, together with the sustainable urban water management. And with this, we can define regional reference diagnostics, intercity comparisons. This is very important also for the mayor, for the city services, to have references of other cities and to have connections with other cities, risk mapping and decision tools, transboundary discussion and practice exchanges, and also awareness approaches and trainings. Okay, now to conclude, I would say this is quite a history. And this is a continuing history of the interface between ecosystem health and OneHealth. Regarding ecosystem health, now we can say that the chemical risks and chemical contaminants has been over time recognized within ecosystem health. This has started in the 1962, especially with the book of Rachel Carson, The Silent Spring, excellent book also with modern concepts in political ecology through key regulations. The REACH is one of them, and this is a critical turning point in the European legislations. And together, OneHealth is now building, starting from the human health. It is an anthropocentric concept first that now has to move toward merging much more the animal first, and this is done already, and embracing and recognizing the role of ecosystem health. And maybe in the future, we can open here to a OneHealth concept that includes micropollutant impacts and socio-ecosystem trajectories. Thank you very much.
Thank you very much. You hear me? Yes. Thank you very much, Gwenaël, for this very interesting talk, sweeping from disciplinary, multidisciplinary, interdisciplinary to transdisciplinary research. And you raise the complexity of the issue encountered in water bodies. So many contaminants with, from different families with different properties and behavior. Which can degrade it into different transformation products. Certain are not well known, and also the cocktail effect, which is much less studied and is still now a little bit challenging. So we can sweep to the question session.
Thanks, Gwenaël. Hello. Thanks for this very clear and interesting presentation. During the 2 or 3 last decades, new pollutants. So, hello.
Hello.
During the 2 or 3 last decades, new pollutants seem to appear, but it's why just they were not taken into account. So, which kind of pollutants not taken into account yet can appear?
I think what appears is what we can detect. So this is a first answer. The invisible is probably the most important part. So I would say what will appear are transformation products. You see, one single molecule will give rise to 20 or sometimes more transformation products. Currently, in most of the world aquifers, we have problem not with atrazine, which is a herbicide widely used, but with desethylatrazines. We have no problems with metolachlor and other herbicides, but with ethane sulfonic acids of metolachlors, and so on and so forth. So I think here maybe new molecules will appear. The question is when the regulations, and here also the European Commission has a huge role to play, will take into account this full circulatory understanding and cycle thinking ways of the micropollutants. What is released cannot be recovered, and we have actually giving the diversity of the ecosystems not a real idea of what it will give once it is released in the environment. I would say the problem is for the antibiotics, new antibiotics and so on. As this is always human first, and for good and fair reasons, we all understand this, it is very difficult to to say, okay, one should reduce, or maybe one should reduce the antibiotics, and this is the way to do it. But eco-prescription is another way to reduce it. Discussing with the doctors, with the veterinary schools and the people there to say, okay, you have the alternatives between different molecules. Some of them are much more biodegradable. So let's see what the future will give. Let's continue to monitor. Let's strengthen the regulations, and let's go for life cycle thinkings.
For the future.
Yeah.
Vincent Durlac, thank you for a very interesting talk. In your talk, you mentioned metformin, which is an anti-diabetic drug. What is the source of it? Is it treated patient? And do we have any idea of the consequences of this, the presence of this compound in human health?
Yeah, so metformin is an anti-diabetic compound. So it's mostly used as a glucagon. This is a really typical pharmaceuticals used by many people, and this is one of the top pharmaceutical. Metformin, we have observed that the degradation of metformin, first of all, is quite complex. It's not one single pathway. It really depends in the environment, and it's quite persistent, actually. It's going through the guanylyl pathways, and guanylyl and some of the transformation products are also persistent. So the main source is wastewater treatment plants, and within wastewater, the degradation will be about 5 to 10%. So that means that you have between around 90% of the metformin entering into the wastewater treatment plants that will go out of the wastewater treatment plants, and that will be rejected in aquatic ecosystems. The consequences, it's under investigations. What we could see here, for example, on the microorganisms, it has an impact on the microbial communities. And we know also that it can sorb or accumulate in some organisms. So this is the point where we are now. Yeah.
Do you think that the REACH regulation have a positive impact onto the decrease of pollutants, even if it doesn't take into account the degradation products of the pollutants?
Yeah, of course, definitely. Without the REACH, we would have much more problems. And what is important also with regulations, as we can see now with the cases of climate litigations, is that, it puts the topics in front. So it allows also to put the pressure and to the citizens, to the associations, to really push these questions, and also to have an impact, I would say, on the delegates, the diplomats, and so on. This is already, REACH was, since the early 90s, was into the cartoons and ready to be launched. But the necessary steps to do this was some scandals. For example, chlordecone or other scandals with chemicals, that was really determinant for launching REACH. And of course, it's not sufficient. But the question is, once we have a regulation, is then how to monitor it. And I think this is also a life cycle assessment of regulations that we should think about. Yeah.
Hi, Laurie Hamler from TBI, Toulouse Biotechnology Institute. I was wondering, when you showed the circular economy, I mean, you spoke about prevention, but there is also the aspect of reusing and therefore concentrating pollutants back into the supply chain, let's say. So I was wondering if that's something you take into account, and especially when it comes into interaction with humans and food products, perhaps, and packagings and so on.
Yeah, exactly. For example, you have two cases, pesticides. How to recover pesticides out of the fields. There are attempts to do this, but this is very difficult. Once pesticides are spread in the fields, part of it will go into the groundwater, other parts will be transported via runoff into the surface water, and this is called diffuse pollutions. So how to concentrate diffuse pollutions and to go through this reuse? It's not really possible. So here for this, we really have to think about why to launch, why to use those compounds in the fields in such amounts, with such frequency, and what could be the alternatives. And this is, I think, really questioning the way to produce the food. And we cannot see only simply micropollutants. It has to be associated with the agroecosystems in which it is, and going back to the food chain, how the food is produced. Yeah.
I mean, I agree. So it's about the future, but what about the past? The past materials that we want to circulate back, what about these?
You mean, for example, for diffuse pollutions, micropollutants are mostly associated with diffuse pollutions. So what is found in the environment, it's not possible, to my opinion, to come back much more. It's released. And this is spread. I do not see ways you can, remove all the sediments from one lake and so on. It's very difficult. But for example, for pharmaceuticals, we have really other ways to think about it. How to reduce, for example, the parts of pharmaceuticals going to the wastewater treatment plants. Using different types of molecules, by changing some few functional groups, sometimes you can have a similar effect on the diseases, but it's really readily much more biodegradable. So this kind of thinking can really help to move and to think about the receptor ecosystems. Then, of course, you have the industrial pollutions, of remediation. Yeah.
Thank you for your presentation. Actually, I have two questions regarding your experiment where you studied the impact of biocides and other contaminants such as metformin from wastewater treatment plant. Could you explain why their co-presence had the highest impact? Like, what's the type of interaction? And my second question is, you just mentioned that the metformin biodegradation is complex, but can you be sure that other contaminants doesn't undergo hydrolysis, for example, in your experiment?
So for the first questions, you are questioning about the mechanisms. Why? Is that correct? Why those? Yeah. Yeah. We think that they are both herbicides. And one of them is focusing on the glutathione pathways, which is also found in the bacteria. So it's more technical, but let's say plants and bacteria are sharing similar pathways. They are not exactly the same, but then it can impact them. And the same for terbutrine, that may touch the photosystems. And some of the molecules involved in bacteria or enzymatic systems is also partly connected with these pathways of photosystems, especially for the photosynthetic bacteria. And for the second questions, I have not understood what you wanted to.
My second question, that you mentioned the biodegradation of metformin is quite complex.
Yeah.
And what are about the degradation of other contaminants? You mentioned that they can be undergo hydrolysis, for example.
Yeah. Yeah, okay, so for the contaminants, it really depends. You have hydrolysis, you have the photolysis, and the photolysis can be direct or indirect, and this is a completely different world. And then, of course, you have the biodegradation. So these are the three main pathways. Hydrolysis will play a role at the extremes of pH. For example, when you are above pH 10 or below pH 2, then you have the impact of the hydroxyls or the protons, which can directly affect the molecules. And it depends on the molecules and the propensity to be degraded by this. For the photodegradation, what plays a very important role in rivers or aquatic ecosystems is the indirect photolysis. Indirect photolysis, that means that the sun will excite dissolved organic material or nitrate in the water, and then the energy will then pass to the molecules. So it's not directly the sun that is breaking the molecules, but it has a cleavage through the other molecules present in water. And of course, this complexifies. You can know what is happening in your beaker in the lab. In the field, you have all these co-occurrent processes, and this is very difficult to know what is the part of each. But this can be done, for example, with stable isotope analysis and so on, also in the field, much more. Yeah.